I'll start recording. Yes, so probably you guys can hear me. Good evening, everyone. Good evening, good evening. Okay, so it's better if I close my video because the issue is like that our network is also not working fine and my laptop is also not working fine. Okay, so that's the thing, nothing else. Yep. So we will directly go to the today's thing, that means metabolites and about the enzymes. Okay. Before we going to start about those things, can anybody tell me what are these objects are? The objects one and two. What are these? You guys think? What may be these things? So any guess or anything? So I'm just waiting for the answers from both sides. Good evening, Gopika. So, any idea, guys? So, what it is? These structures. So, if you know, just let me know if you guess anything. Otherwise, just put that I don't know. Like we don't know. Okay, so I can explain. Exactly, I got some answers. Correct, Jivan. Not 100%, but correct. Okay. Anything else? Good evening, good evening. Exactly. These are the protein conformations. Okay. Protein conformations. Conformations or structures, all those things are same only. Okay. But this is actually an enzyme. Okay. Enzyme one, we can state we don't know the name of the enzyme. That's why we can keep this as the enzyme two. Okay. If you take this particular example, okay, so you can see that this is a kind of an enzyme, right? It's showing a three-dimensional nature, right? So if you see in this particular structure, okay, if you see in a proper way, here you can see alpha helix also, right? Alpha helix structures here you can see here and there. Okay, here you can also see the beta sheet, but this is not proper in here, so we will go to the second enzyme. Here what you can see here, right, this is the beta sheet. So look how it's arranged parallel, right? And this one is the alpha helix, right? This one is also alpha helix, this one is also alpha helix. If you can start it from here, just observe it. This is the single line, right? This is the single line. They simply change the color to represent the different portions, okay, to represent the different portions here. So as you can observe here, all of this belongs to the somewhere comes to this again comes to this, right? So this is a single polypeptide chain which is in a tertiary structure. So this tertiary structure is made up of the alpha helix also and also having a beta sheets, okay, also having the beta sheets. So I think you guys understand how the conformation is formed, right? How the conformations are formed. So it may be a single kind of a thing, maybe formed of a single helixes, or maybe formed of the sheets, or maybe formed of the both, right? If there is more than one polypeptide, then it will become the quaternary. Okay, so what we studied in yesterday class we studied about the different polysaccharide types, right? So it may be a homopolysaccharide or it may be a heteropolysaccharide depending upon its components or the monomers. And what are the other things we studied? The other things we studied are, so these polysaccharides may form through a glycosidic bonds, right, by the condensation. And what else we studied? We also studied about the nucleic acid and the components DNA and RNA and types of the RNA molecule and how the bondings is happening and what's the meaning of 5' and 3' and all those things. Okay, so we will move on. The one bad thing about today's class is like very few questions are there. Okay, the only reason is like this laptop is not working. Whenever I try to do something, it's going to some other pages. So I can't edit any PPT. I left it like how it is yesterday only. Okay, so no changes happened here. The first question came. Okay, the question is simple. in a multiple intracellular processes, that means there are several processes going on. Okay, one or two, we don't know. Okay, DNA is giving rise to an enzyme amylase. What's happening in a process? DNA is giving rise to an enzyme called amylase. Okay, and they're asking, then find the components of the enzyme. Then what may be the components of the enzyme they're asking? Okay, the options are option A nucleotides, option B deoxyribonucleotides, option C amino acids, and option D monosaccharides. Okay, I'm just launching the poll. Who are in the Zoom can answer in the poll, and who are in the YouTube can answer in the YouTube live chat. Okay, so good evening, Ashwin, and others also. Like, is anything special today? Like, attendance is like too low, right? What happened? Anything is going or any exam, test, or anything, any school? Because like it's almost 40% decreased. That's why. I'm getting so much of the complex answers. Okay, we will come back to that. No issue. I think for every student, biomolecules chapter is done in the CBSE, right? Is any school is there in which biomolecules chapter is not over yet on 21 unit test? Okay, 21 unit test you have. This is like long gap. No issue. Art integrated projects are given. Okay. Huh. Jayant, it's not going on. Good evening, Naina. Hassan, it's going on, right? Huh. Yeah, I got it. Okay, I'm ending the poll. I think you guys got a lot of time, like more than enough time. Okay, I'm ending the poll and I'm sharing the results. 16% chosen option A, 47% B, 18% C, and 18% D. I'm just waiting from YouTube till. Okay, I'm stop sharing. Okay, so we'll come back to the question then. The question is simple. Uh, Amit, you will get to know, okay, how you can find the answer. So they're asking in a multiple intracellular process. What's the meaning of intracellular? That means within the cell, right? Within cell, there are some processes going on. We don't know the process, and the questioner don't want us to know the process. Okay, we'll leave it. Okay, some process is going on. What is happening in that process? DNA is giving rise to amylase. Okay, amylase. The amylase is an enzyme. Okay, amylase is an enzyme, and they are asking, you have to find out the component of the enzyme. You have to find out the component of the enzyme. Okay. Okay, like it's like a blind question, it seems. Okay, I will ask some other questions then. We will keep in this one here. uh, from what kind of molecules enzyme will be formed? Can anybody know that? Do anybody know that? Because for some of these schools it's already over, right? Correct, Ashwin. So from what kind of the molecules the enzyme will be made, or from what enzyme will be formed? Can anybody answer to this question? I'm just waiting. Exactly, my Shapa, Abhishek. Correct. Anybody else? Only two answers I got. Likita, correct. Prakriti, Sinchana, exactly. Exactly. What are enzymes? Chirant, exactly. Deepika, correct answer. And I'm waiting. Yeah. So everyone is correct. The enzyme is made up of proteins. Okay. Even though not all, we will come back to that one time. Okay. In the next slide, I will go to that. Okay. But most of the enzymes are proteins. Okay, most of the enzymes are protein. So okay, we come to know that enzymes are made up of the protein. Okay, then we will come back to the question one more time. That means they are asking what is the component of the enzyme. We can write it down like this, right? Components of protein. right? Did you guys understand? So DNA is giving rise to amylase through a process. We don't know what process it is, okay? But we do know the enzymes, amylase enzyme they mentioned, and enzymes are made up of the protein. Most of the enzymes are made up of the protein, right? So we can change the question to something like components of enzyme to the components of the protein, then and everyone can answer, right? What are the components of the proteins? Which is the correct answer? Component of the protein. Component of the protein will be the exactly amino acids. So the correct answer is amino acids. Remember, sometimes you will like struck with something which you don't know. Okay, like in the first statement, it's simply they gave. Okay, the first complete statements are simply to distract you, right? It may be a multiple intercellular process or direct process, whatever the process. DNA is giving, RNA is giving, we don't know, we won't want it. The ultimate thing is the enzyme is made up of the protein. That's the ultimate thing, right? So if you want to know what are those intercellular processes, I will simply explain it here. No need to take tension because we will learn these things later. Okay, DNA will goes through a process called transcription. Okay, transcription. So it's a kind of process, a multiple step process, and this DNA will give rise to the RNA. Okay, specifically mRNA, but we will keep it like this. Okay, this RNA, I will no. Then both options will be correct, Abhishek. If the options are protein and amino acids, then both options will be correct, right? So that will be a grace marks, right? So this RNA go through another process called translation, and this will give rise to the product of this particular process, that will be the protein. Okay, so after this protein we got not protein, actually we will get the polypeptide chain, like polypeptide chain, something like we will get. Then the foldings will happen. Okay, protein folding will happen and it will give rise to our enzyme. Okay, our enzyme. Can anybody tell me where actually the folding is happening? Okay, we will get a protein after a process of transcription and translation, right? From the DNA to RNA transcription, RNA to protein translation, we got the protein. Then folding is happening. That folding will give rise to the enzyme. Okay, so where this folding is happening then? Can anybody tell me where this protein? Like, I'm not asking how the folding will happen. I'm asking in which component of this cell, which component of this cell the folding is happening. Okay, where it's happening. Like I'm asking where this gold necklace is come from. Okay, where it's manufacturing in the jewelry, right? Exactly. So similar manner, I'm asking where this enzyme jewelry is happening. Exocrine gland, membranes, ribosomes. Okay, any more? Okay, correct, Mahesh. Not 100%, but most correct. Correct, Mohan. Most correct. Like, did we mention anything related to that in cytoplasm or mitochondrial plasma? Then, no, we mentioned the protein folding in only two phases, if you remember properly. One in the endoplasmic reticulum, exactly, Sinchana, right? One in endoplasmic reticulum, another one is the Golgi bodies, right? We mentioned what happened in endoplasmic reticulum, especially in rough endoplasmic reticulum, right? So the protein will be secreted, that will be transferred in the endoplasmic reticulum, vesicles, tubules, and everything. Their protein folding is happening. Then sometimes we have to add some other groups or some other things to the protein, or we have to make it 100% correct. That process will be happening in Golgi, right? Only two organelles which are helping in the folding of the enzymes, sorry, folding of the protein, that happens in the endoplasmic reticulum and Golgi. Remember, when protein is formed, is simply a linear chain. Okay, simply something like this. Okay, but after it's passed to the ER and Golgi, okay, especially in ER, remember, these ER and Golgi molecules turned it into different shapes. Okay, turn this into either a helix or a beta sheet or anything like that. Okay, so remember about this. So where it's happening in there. Okay, I will move on then. So today we are discussing little different. We already discussed about these things. I'm just showing the things. What are the monomers and what are the polymers? Okay, so you can check these are the monomers which are building this kind of the polymers. Okay, this kind of the polymers. The amino acids are the polymers of the protein, exactly Gopika, and which is with what these things are with the a peptide bond, especially with a peptide bond, other kinds of bonds also there. Okay, second, the fatty acids. Fatty acids is one of the monomer of the phospholipid. Okay, and glycerol is a kind of the carbohydrate. Okay, alcoholic carbohydrate you can see. Okay, a carbohydrate with a alcohol functional group you can say. Okay, so this is also one component. When both of them joined, they may form a phospholipid or they may form a fat. Okay, or they may also form some other kind of the lipids, complex lipids. Okay, so that's what they mentioned. And carbohydrates, in that monosaccharides are the monomers. Mono, you can remember that. These will combine to form either oligosaccharide or polysaccharide. Here I forgot its ester bond, and this one is the glycosidic linkage or glycosidic bond. Okay, and the last one is the nucleobases, and nucleotides are the monomer. So how these nucleotides are formed? There is some given away from the monosaccharides also. Can anybody tell me which is the component which is given from carbohydrate to the nucleic acid to make this nucleotide? Exactly, ribose sugar. Ribose sugar, right, which is given from monosaccharide to here, and when it's joined it will form a DNA or RNA depending upon this particular ribose sugar. Remember, okay, this is how the process is happening, right? Wherever a monomer is there, ultimately this monomer is converting into a kind of the polymer. Okay, and whenever a polymer is there, like if I give you an example, if I give a biscuit to you, okay, biscuit for you guys to eat, the biscuit majorly contains sugar, right, sucrose, and it's also contains starch, right, and it may also contain a little bit of oils. So what happen in your digestive system? In your digestive system, one second. In your digestive system, what happens? It will be broken down in formed into a monomers, right? It will not kept it like that only. So in your digestive system, a starch is broke down into glucosinate, a sucrose is broke down into glucose and galactosinate, sorry, glucose and fructosinate. Okay, oil is broken down into other kind of like glycerol and the fatty acid molecule. Okay, and a protein can be divided into amino acids. Okay, so when your body takes that, it again reutilize itself, right, again reutilize all those things and to make the another kind of the this one polymer. So this is what we call, right, catabolism and anabolism. We will go through that one more time, but we, after so there is no question, sorry, I forgot. Yeah, so after this, okay, so that's what we are discussing in the first class also, right? What is metabolism then? Metabolism is the complete state or complete reaction what is happening in your body, right? Complete metabolic reaction, sorry, complete biochemical reaction happening in happening in living living system, right, happening in a living system. It may be in a cell or it may be in you, right, it may be in a bacteria, that doesn't matters, right, wherever it is happening a metabolism is happening. How it is happening? A larger molecule is there, it is going through a process called catabolism, and this catabolism is giving rise, or how can I say, it's a breaking down, and it is forming the breaking and forming the smaller molecule, and this smaller molecule again going for joining. Okay, joining a kind of the things one to one join, and it will form the larger molecule. Okay, during this process, actually energy's production is happening. Energy may be in the process of ATP or in the some other kind of the molecule, NADH or FADH. All that things we will learn later. Okay, no need to take tension about that. These are also like ATP only. Okay, NADH, FADH also, but majorly ATP or ADP molecules we are getting, right? So this is what metabolism is. So when you're going through the metabolism, like two pathways we normally observe. One is the anabolic pathway and a catabolic pathway. I'm repeating it one more time. Okay, so because this is simple, but this question normally asked. Okay, that's why. Anabolic pathway means simpler molecule will form the complex molecule or complex structure. So you can call constructive process, like a construction of the building, right? We will take cement and we will take bricks and everything. We will construct a building. That's the anabolic pathway, and it consumes the energy, right? For building a this one, for to build a particular kind of a building or house, we need energy, right? We have to eat, we have to push everything, and we have to place it in order. We need energy, right? Try to think about a building. So example is like, from acetic acid we can get the cholesterol, okay, by the different process. From the amino acids we will get the protein, okay. Photosynthesis we will get the glucose, right? All this where this glucose will come from the CO2 and H2O, right? The CO2 and H2O will form the glucose. All these are kind of the anabolic pathways. But catabolic pathway is from complex molecules to become simpler molecule. That means destruction is happening. I already built a house or I already built a building and I want to destroy it, okay. It releases energy. Remember, if you destroy a building, okay, you put some bomb or something and the building is collapsing, okay, then lot of amount of energy will be released into the nature, right? The other buildings will shake, the earth will shake, and it will produce the sound energy and everything, right? Why it is? Because it's releasing energy, not consuming. But when you are building the house, the sound is less, right? Because it's consuming the energy. But when catabolic reaction, suddenly, abruptly, it will release the energy, okay. Exactly. The examples, glucose may divide to form a lactic acid. Okay, glucose is the first molecule. A different enzyme and different process will happen, which give rise to lactic acid molecule. Okay, that's what we called as like regular process. Exactly, Ananya. And through the respiration, what happens? We will take a glucose and everything. By utilizing the oxygen, we will destroy it, that or we will divide the glucose or any other molecule, and we will get the energy, right? So whenever a catabolic pathway is going on, it normally releases the energy. Whenever the anabolic pathway is going on, it's actually consuming the energy, okay. This always happen in a cyclic manner. That is very, very, very important. You have to remember that, okay. Last slide. Simple, this much only. Last slide, nothing else, okay. So industrial is exactly. So remember about it. So whenever these two things are happening in a cyclic way, okay, if you stop any one of the process, conversion of any one of the process, it will lead us to the disaster, okay. I will give you the example. Think that you are stopping anabolic pathway in your body, okay, anabolic pathway in your body. That means what happens? You will instantly die, because for you to live you need proteins, right? And you need enzymes and you need other molecules. It's not happening, right? What happens if we stop our catabolic pathways? Then also we will die, okay. Why it is then? For us to live we need energy. For that catabolic reaction have to happen, right? So both the reactions should be there in our body, and both the reactions should be happening simultaneously to for us to live, okay. That's a very important thing. So we will move on. Then what are the substances which are involved in these things, okay. So the substances which are involved in metabolism is called metabolites, okay. What are metabolites? Metabolites are the substances involved in metabolic process. Substances involved in metabolic process, involved in this kind of biochemical process, I can say. Biochemical process, okay. So we divide that into two types. Simple, okay, two simple. Primary metabolite and secondary metabolite, okay. What are primary metabolites? Hi Akil, you are late. Yeah, primary metabolites means The substances which use it to gain energy, substances which used to gain or use it for growth or to gain energy. Okay, use it for growth and metabolism. So I will give you the example, then you can understand. Okay, the example of the primary metabolites is, as you guys already know, a carbohydrate and a protein, okay, and a lipid. Okay, all these things are primary metabolites. Okay, that means what happens when you take a carbohydrate or when you take a protein or when you take a lipid. Okay, so what is happening, those things are helping in building your body, right, or sustaining your body, right? So it may be visible or not, but it's help in the deposition of the body. Those are called as the primary metabolites. We normally say, right, these are primary important thing or primary thing we have to do. Primary means what? Prioritizing things, right? That's very, very, very important. So those are the primary metabolites for the growth. Okay, what are secondary metabolites? Secondary metabolites are substances which are not primary metabolites, or in other way, which are not involved in growth, which are not involved in growth. Then why it is useful? Then it is not useful for growth means for what it is there. Okay, but helpful helpful for defense and other things. Okay, defense and other things are the important here. Okay, so once again guys, give me 1 minute of time, I will back again. Okay, some problems happening. Yeah, I think everything's sorted. Okay, sorry for the like small distraction. Sorry, disturbance. Okay, so secondary metabolites are what? Which are not directly involved in the growth, but helpful for the defense and other things. Okay, what is the meaning of defense then? So it may be helpful against a bacteria, or it may be helpful against a fungi, or it may be helpful for some other things. Okay, we will see what are those things. So if I give you the example, it may be a steroid. Okay, or it may be some other things. Okay, antibacterial substances, these things. Okay, remember, you have to remember this one thing: these primary or metabolites, primary or secondary metabolites, especially produced in the plants or especially produced by these metabolism in this metabolism. Which metabolism? I didn't understand. This means what? Okay, so primary and secondary metabolites both are produced in autotrophic organisms only, and heterotrophic organisms will utilize that. Okay, if you take care of the secondary metabolites only produced from the plants or the autotrophs, okay, not the other things can. Plants means plants, fungi, all those things only can produce these things. So we will keep it in this way. Okay, except animal others produce, or you can also keep it something like except heterotrophs. Okay, except heterotrophs other will produce. Okay, so these things will be produced from the autotrophs or in the plants, then us, like all the other things, will take this one. Okay, we'll take this one and we will utilize those things also. I will tell you, like we have some example, but fungi also produces. Fungi also produce the secondary metabolites, but not the primary metabolites. It's also go through different kinds of the process. Okay, different kinds of the process. Exactly. We will see some of the secondary metabolites here. Okay, we divided secondary metabolites into different categories chemically. Okay, so chemically we divide into several categories. If you see pigments, okay, pigments means which give the colors. It's a secondary metabolites. It never useful for growth. Okay, So example are carotenoid or anthocyanin, the colors which is given to the flower or the fruit and to the some of the tubers also. Okay. Second are alkaloids. Alkaloids are also a kind of the chemical structures. It's a aromatic structures. Okay, alkaloids. So examples are morphine and codeine. Normally, when you are having too much of pain, some people will take anacin, right? So what is the component in anacin? It's a low base of the morphines. Okay, so these are some of the chemicals which are actually helping reducing the pain. Okay. And terpenoids also a long chain or multiple ring structures. Okay, these help in like our body to participate or body to active be in active stage. Okay, some of them are essential oils. Essential oils are the things which can be obtained from some other plants or other sources. Okay, which will be helpful in rejuvenating or recreation of the our body. Okay, some of the toxins. Okay, toxins, resin which can be obtained from our what is that, a resinous communis plant. I'm not sure about what it is saying. I forgot the common name. Okay, resinous communis plant. So this particular is against used in for the killing of particular animals. Okay, lectins also are intermediate metabolic things which actually attach to a particular kind of the carbohydrates. Example is concavalin A. Some of them may be drugs. Okay, drugs means which can be useful for some diseases like vinblastine, which can be used for cancer. Okay, a curcumin which can be used for the antifungal or antibacterial. Okay, or maybe a polymeric substance like rubber, gums, or cellulose. Okay, so all of these things are secondary metabolites, and some of these are examples. Remember, none of these things will help in the growth of the plant. Okay, none of these things are helping in the growth of the plant. Then why it is useful? All these things are useful against the defense, against the bacteria, fungi, or any herbivorous animal. Okay, some of the plants will produce the toxins. Okay, what's the reason for that? To protect itself from others to feed on, right? If it is like a regular plant, then what happens? The deer will come, the cow will come, the buffaloes will come, or humans will come, and we will eat, right? If it produces a toxin, nobody will touch it, right? So it's a helpful process, right? Like that, probably it will produce. Or some of them produce like curcumin produce, this one, our which one we will take? Yeah, curcumin is there, on vinblastine is there. Why it's producing those substances? To kill some other bacteria or some other pathogens. Okay, it's not producing all these things for us, it is producing all these things for itself. Okay, we are utilizing this chemical in different way. Okay, why you like we you normally drink tea, right? So it has some kind of boosting kind of the things, we will say. When you drink the tea, you will get the energy and everything. Where the energy is coming from? The energy is not coming from the sugar, or it is not coming from the any kind of the primary metabolite. It is coming from the secondary metabolite which is present in the tea, right? Tannin or anything, so which is actually, or caffeine, which is actually boosting your mind. Okay, it may be a steroid. Sometime some people will take this illegal drugs, right? Those drugs will be coming from the plants only. Or you will take nicotine from the where this nicotine how this nicotine will be useful? Is the nicotine is a primary metabolite? No, nicotine is a secondary metabolite. Plant produce the nicotine to destroy the insect. Okay, nicotine is an insecticide. Remember, nicotine, what we normally see, right? Nicotine is an insecticide. That means the chemical which kill the insects. So plant produce the nicotine to stop the insect attack. Okay, but we utilize that exactly same nicotine for our recreational purpose. Okay, so remember, so secondary metabolites are not produced in humans or any animals. It will be produced in plants or some other things. We will reutilize in different ways. Okay, so if drugs are produced from plant, then why it is illegal? It is illegal because these things, we humans can like misuse it, right? If you take an example of a morphine, right? Morphine, or if you take an example of codeine, or if you take an example of opium. Okay, where nicotine is used. Nicotine is used in your what is that? What do you guys call it? Supari and everything is there, right? Vimal, pan masala and all the things. It contain the nicotine, right? Now our nicotine, uh, uh, what is that? Tobacco. Tobacco contain nicotine. That's what I'm talking. The tobacco plant is producing. I will come to that right now. Okay, what is that? So opium is a potent drug. Okay, morphine and opium can reduce your this one paining sensation. Okay, so you can utilize those things medically to reduce the pain or symptom of the patient of having some injury or anything. Okay, it is actually used in the Second World War time, morphine and used. Okay, but if you take that in a high dose, you will go to a trance kind of the state. Okay, so you can dream and you can have like you feel like floating, hallucination will happen. Some people will enjoy that, so they can grow and sell it to various kind of the people, right? Then society will broke down. That's why these things are illegal. Okay, and cocaine. Cocaine is also a secondary metabolite, as you can see here. Okay, so probably it's not mentioned in here. Yeah, cocaine, opium, everything. Okay, so nicotics. Nicotics is a tablet, right? Like to stop the nicotine addiction. That means tobacco addiction, especially for the cigarette addiction, right? So cigarette also contain the nicotine. So what nicotics contains then? Nicotics don't contain anything which is against this thing. It also contain nicotine only. Okay, what happen, instead of they are smoking cigarette, they will take the same amount of nicotine in the form of this chewing gums. Okay, then they will leave the habit of smoking, right? Then they can reduce the habit of this tablets also. That's how. You can't stop something some drugs directly. Yeah, because if you stop some drugs, that particular patient will go for a withdrawing symptoms. Okay, withdrawing symptoms will be very dangerous. Like think about some person who is drinking alcohol from like 4 5 years or some several years, and you make him stop drinking from that particular day. Okay, then his body is wanting that because his body is adjusted to that, right? It's started to act in a different ways. He may try to like lose himself, or he may act like a madman, or he may try to kill himself. Okay, all those are withdrawing symptoms. That's why instead of stopping it directly, we'll go through a course plan. That's how we will start to reduce it. Shivering, all those things will be there. Okay, drugs harmful, yeah, exactly, but not everybody will be like us, right? So like if something will give you money, so we'll go for that. We will not at all care about some other people's, right? So who cares for the others? Like we need money and we will get the money. That's what happens, right? Curcumin is turmeric, exactly. Curcumin is from the turmeric. Thank you, Tejas. Yeah. uh, and what's another thing? So some, as you can see, carotenoids from the, not all of them are drugs. Okay, in this secondary metabolite, some of them are drugs, some of them antibacterial, some of them antifungal. Okay, that means destroy fungus, destroy bacteria, or helps to boost your immunity. Okay, if you take a carotenoid or anthocyanin, those kind of things, or if you take some other things, flavonoids. Okay, flavonoids in tomato and all the things, it will boost your immunity. Okay, so morphine, codeine, those kind of drugs. Or eucalyptus, we normally use it for aroma, it's an essential oil. Okay, eucalyptus oil. Or cinnamon, we normally use it for biryani and all things, right? And cloves also for the flavor. Ginger for the aroma, or ginger for the oil. Okay, different color flowers and everything for its smell. Okay, so we'll use. Okay, what's the polymeric substances? Polymeric substances are formed from several monomers. So the best examples are rubbers. Okay, so whatever we are using, the rubber which can be seen in different process like earbuds or anywhere, this is coming from the rubber plant, right? Rubber plant. So rubber plant will secrete this rubber to stop bleeding. Okay, just take a plant, and you don't have the work, then what you will do, you will cut here. Okay, you will cut somewhere here. What happens, that this will expose to outside, right? Then bacteria can come here and sit, and it can kill the complete plant. To stop that, what these plants will do, this will secrete a kind of substances which will fill this hole. So bacteria can't come here. It can be bacteria can't affect this plant, right? So those kind of things are rubbers, gums, or cellulose. Okay. So gums, it's also in the similar ways. There are like several plants which can produce the kinds of the gums, not the regular gums what you can see. What you rubber gum, sorry, chewing gum, everything is actually now commercially we are actually producing artificially. Okay, but naturally these gums are produced from the plants only. Okay, and lemongrass oil, there is a grass called lemongrass. Okay, so it's also like a regular grass only, but this grass is having lemon type of the smell. Okay, probably some of the people already seen it. I seen in several JNVs. Hassan people probably know it because in Hassan JNV we can find here, right? Near to the Aravali house, right? So this will having the a lemon kind of the smell. Okay. In ancient time, we normally make lemongrass tea with this, especially in Japanese. But we come to know that we can also extract the oil, essential oil, which can be helpful in several ways. Okay. Even you've seen the Navaratna Taila, right? Navaratna Taila, what we normally use. From where this Navaratna Taila is coming from? Navaratna Taila is comprised of several kinds of the essential oil and the medicines. Okay, especially some kind of the tuber oil and all the things. Okay, gum is like the color of honey now. Not always, not always. Gum may be in any color. Polysaccharides are heteropolymer, but cellulose is homopolymer. No, 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 no. Like, really? Did I told cellulose is a heteropolymer? No, I haven't told cellulose is a heteropolymer. So just check once. Okay, just check once there, Chirant. Okay. So what about cyanide? Cyanide is also toxin which can also be produced in some of the other things. Okay. So that's also produced to stop plants to go through the, okay, to stop the herbivory. That means the it stop other things to eat that particular plant. Okay, no, we didn't mention the cellulose in here, heteropolysaccharide. No, we mentioned in homopolysaccharide. Check, Chirant, one more time. Okay. So cyanide is also produced in the similar way only, but now we are using it for the different purposes. Okay. There are like, even though several different kinds of cyanides are there. Sorry, I went back to check something. Okay. So pectin, pectin polymer. Pectin is a composition. Okay, so a kind of polymer, you can say exactly, but it's not a simple polymer. It's also have, we can say, metallic. A kind of polymer which is also comprised of some other kind of the molecule. Yeah. So one question came. I will search you in 1 second. Okay. So coffee plants store high level of caffeine in seeds because or for to taste good, to recreational purpose of the humans, to avoid pathogens, to reduce the germination rate. Okay. Germination means, from seeds the plants will come. Reduce means to decrease. Okay. I'm just relaunching the poll. You can answer. So exactly. You are asking why cyanide will cut all ways it goes. The simple thing is cyanide is a high level coordinating compound. Okay. You guys know about the coordination compounds. Probably it will come in the 12th class. I think, right? Coordination compounds. So cyanide, what it will do, it will start to form bond with all the things. Okay, start to form bond with everything, especially with the hemoglobin. Okay, when it starts to form bond with the hemoglobin can't bond with the oxygen. So it will cause the reduce in oxygen level in the blood. We normally call it anoxia. Okay, so anoxia means there is no air. Okay, so or there is no oxygen. Okay, so this anoxia will leads to the death of the brain. Okay, when there is no amount of oxygen is supplying to the brain, so the brain will start to die. Then all the other body parts also start to die. It's not reversible. Okay, it's irreversible. That's why that's very dangerous. Plastic is also polymer. Then how it is different? Plastic is an artificial polymer. Okay, so what we discussed here is a natural polymer. That's the only difference. You will also learn the polymer in the class 12 chemistry polymer chapter only. Okay, so polymer may be artificial or natural. For artificial you can go for PVC or plastic. Okay, all those things. But natural, these things, gums or this one's rubber, all those things. Okay. I'm ending the poll. Yes, exactly. I'm sharing the result. 10% chosen option A, 7% B, 67% C, and 17% D. And in YouTube also, I got the answer C only. Exactly. Okay. So why coffee plant have the caffeine, or why coffee plant store the high level of caffeine in seeds? Not for us, okay, not for the us, or not to reduce the germination rate. It should germinate properly, right? This is also not the correct answer. The correct answer is to avoid the pathogens, okay, to avoid the pathogens. Normally, if you observe regularly, okay, lot of seeds are poisonous, okay, compared to the plants. The seeds are poisonous. If you take the abrus, like what we call gulaganji, we normally call it, right? I don't know what it's called in English. I know the scientific name in Kannada name, okay, abrus precatorius or gulaganji. So those seeds are very very poisonous, okay, it's a high poisonous. So but the plant is not poisonous, then why it's only seeds are poisonous? Because seeds are the one which will give rise to the another plant, right? We will not, we will know it. Exactly, Jayant. We can go for this apple seeds. Apple is not poisonous, apple plant is not poisonous. You normally eat the apple, but apple seeds contain cyanide, a very low amount of the cyanide, still it contains a cyanide, okay. But that amount of cyanide is enough to kill an insect, even though it's not enough to kill you a single seed, but it's enough to kill the insects which normally try to feed on the seeds, right? The plant ensure, I bet it longer one, no problem, one or two seeds nothing will happen, okay. We are, everyone of us probably already eaten, right, one or two seeds of the apple. The plant have to ensure that the seed should germinate, right? But what happens, a lot of times the insects will attack the seeds and it will destroy it, right? To stop that, normally plants will store the toxins in the seeds, okay. Seven seeds, the like not, no, that's not mandatory, it always differs, okay. Not like seven seeds to enough to kill, you are saying, okay. Gulaganjia. I'm not like referring that. Okay, it's dangerous. It's highly dangerous. Exactly, it's dangerous than apple seeds. Okay, there are like a lot of plants are there which is really dangerous. So if you, I will show some one or two photos in the next class. Okay, some plants are there. What happens if you eat those plant leaves also? Okay, PVC of electric use of polymer, electric use. I didn't understand. Like we can create bags from the natural substances also. Okay, from the cellulose materials and all the things. Like paper bag only a kind of things, right? Exactly. So then we come to the living state. What is living state then? The living state is the non-equilibrium steady state to be able to perform the work. Okay, so living state means what? Living state means the a state of non-equilibrium steady state. Okay, remember in physics what happens? Normally all the molecules want to go for the equilibrium, right? Even in the chemistry, like chemistry, physics, everything is same. Ultimately thing, the molecules want to achieve the equilibrium, right? What happens if living system achieve the equilibrium? If living system achieve the equilibrium, then the living system will die, okay. So equilibrium means death to the death to the living organisms. So there is no equilibrium for the living state, remember, okay. So what's the other thing then? The other thing is the living state will always try to be in the non-equilibrium state with a kind of combinations of thousands of reactions. Okay, there should be something is happening somewhere, and some other things will be happening some other ways. Okay, if any instances, if you take your cell, if any instance in if any microseconds, a thousands of reaction will be happening inside your cell, and each of them are independent of other. Like some of them are dependent, but some of them are independent, but none of them are interacting one another. Okay, so they are interacting each other in a separate ways, and reaction will be happening, and they are trying to find a state which is non-equilibrium. Okay, chemistry inversely proportional to the biology. That's what I told, right? So we can't put the same ideas of the physics and chemistry here. That is what made biology different, and that's why biology don't have the common rules. Okay, If it is following, it is possible to go to equilibrium state, but if you go to the equilibrium state, that means death. Okay, death of the living system is equilibrium. Okay, yeah. So that's why, like, we can't put any common rules for the biology, because the living system is so diverse. None of the these things will hold. Okay, none of the statement will hold. In chemistry, we can find one or two exceptions. Okay, or some exceptions, but biology, most of the living system work independently, right? So we can't say it's exception. We can't give any common rules. Yeah, in lesson also it given exactly. Okay, so that's why we are mentioning that this is the living state and this is the metabolites and metabolic things. Okay, remember about these particular chart, okay, which is given in the textbook only, because last time I don't remember which exam they asked about concavely in A. Okay, they asked about concavely in A belongs to. The options are something like lectins, alkaloids, something like that. Okay, that's why the remembering this chart is very important. Okay, polyvinyl, a polymer, exactly. Poly, the name is only have, right, polyvinyl chloride. Vinyl chloride is a monomer. When we join those things, so it will become polyvinyl chloride. That means PVC. Okay, so which normally used in the pipes. So you will see that in chemistry. Okay, a polymer chapter is there, a really interesting chapter. Okay, there you will study about this. So we will come to the other one important thing. That's why we will get more marks in Bayana. Sometimes you will write like in between statements, right? For now, no issue. For NEET, that won't happen. But in regularly, it's a good issue. But later, you have to specify. Okay, if you go for like BSc or MSc, when you wrote some statement, you have to mention in examples for some teachers at least. Okay, so otherwise, they will, if you write a random statements, they will directly give you 0 marks. Okay, some of our teachers, like professors, will directly tell us like, don't write the stories. Okay, don't write any stories or anything. If you know, answer it, otherwise don't answer it. Okay, if you write the stories, they will directly give the 0 marks. If you know there is something is there, if it have some diagrams, then only they will give marks. Okay, that will happen when you go to the medicals or when you go to the BSc or MSc levels. Okay, so here everything sab kuch chalta hai idhar. Right, so everything will run exactly. So we'll come to the enzymes. So what are enzymes? Okay, what's the capabilities of the enzymes? Okay, that's what we are discussing. This is the structure of enzyme. So they're represented in another way. Okay, we will see the first one. What are enzymes then? Enzymes are organic catalyst. Catalyst means what? Can anybody tell me what's the meaning of catalyst? So if you know the meaning of catalyst, then everything will be sorted for us. So probably in several chemistry reactions, you already use the catalysts, right? So increase the rate of reaction, exactly, half statement, exactly. Increase the speed of reaction is a half statement. Where is the other half statement? Increase the speed of reaction is a half statement. Where is the other half statement? Meaning of catalyst. Indirectly help in reaction, directly help in exact. Those things will be there. Exactly, Swati. Exactly correct. Swati, Kirtana, Likita. Okay. Catalyst are the, what are these substances? Yeah. Increase the rate of reaction, the rate of reaction without involving, right, or changing. So if you add a platinum for a reaction to catalyze, okay, even after the reaction you will get the platinum only. You won't get some platinum oxide, platinum dioxide. You won't get something like that, right? You added a platinum, you got the platinum, but you added to a kind of mixture, or you can say reactant, and you got the product, right? So catalyst is the one which never involve or won't change. Okay, but it will speed up the reaction some other ways. We know, we will try to see how it will. Okay, but enzymes are the organic catalyst. Okay, enzymes are the organic catalyst which catalyze the biochemical reaction without being utilized themselves. Okay, they never get utilized, but they will be useful. Okay, so some other statement we will see. Enzymes occur normally in the colloidal state. Okay, what's the meaning of colloidal? Can anybody tell me this one also? Because all this uh, words we normally use in chemistry, right? Probably you guys already know about these things. What's the meaning of colloidal? Colloidal. So, like simple, not completely soluble. Okay. Any other statements? Colloidal meanings. Distributed. Exactly, Mahesh. Any other things? Like I don't think this is difficult, right? You can see diffract light, you can say colloidal is different, not clearly visible, heterogeneous mixture. Exactly. So not completely soluble kind of the things, right? Not completely soluble kind of the things. When light penetrated it, then what happens? It will redistribute, or what it will redistribute the light rays. We have the different laws for that. There are like different machineries working on the same principle. Okay, there are like some laws are there. Okay, exactly. So our enzymes also present in a colloidal state, not in a soluble state. Remember, this is very important. This is present in a colloidal state and often produced in an inactive form. Okay, normally what happens, enzymes are not at all produced in active forms. Normally what happens, it will produce in an inactive form. We normally call it as the proenzymes or zymogens. Okay, proenzymes or the zymogens. Then how it will be activated? Okay, these proenzymes or zymogens will be activated and formed enzymes through different materials. Okay, it may be acid which is activating it, or maybe a metal ion. Okay, or it may be a basic thing, or alkali. Okay, or it may be a temperature. Depends. Okay, depends upon different external things, it will get activated. Okay, there are several reasons for that also. Okay, if I take a common example, uh, take an example of salivary amylase, guys. Salivary amylase, which is present in your mouth, right? Which is present in your saliva. Okay, so what happens, it will divide the starch into the sugar, or the common this one, right? Regular or simple sugars or simple saccharides, right? But that salivary amylase only work in a particular kind of the pH. Okay, so those kind of situations will be there, or only activated in particular pH. If there is like a different kind of the pH, it won't activate. Okay, same thing happens with the different enzymes which is produced in your liver, sorry, which is produced in your pancreas. Okay, when those enzymes are produced in a pancreas, it's in an inactive form. When it will come to the intestine, then only it will get into the activated form. Who already completed the digestive system chapter probably know about those enzymes also, right? Who already completed digestive system chapter know about those enzymes, right? So we will leave that, we will learn later. But remember, the enzymes may be produced in an inactivated form. We call it as the proenzyme or the zymogens. Okay, proenzymes or the zymogens. Due to the external factor, they will get activated. Okay, with a few exception, all known enzymes are protein. Remember, 10th, not about 10th, I'm talking about the 11th digestive system chapter. Okay, so except some exception, most of the proteins, most of the enzymes are proteins, but all proteins are not enzymes. Remember this word also, this is very important. Most of the enzymes are proteins. Okay, with few exception, I can simply put this like most enzymes are protein, but all proteins are not enzymes. You have to remember that the protein may be albumin, or maybe a collagen, or maybe actin, right? None of them are enzymes. But if you take enzyme names, it may be amylase, or tryptokinase, or heterokinase, or kinase, or what's oxidoreductase, ligase. Everything is the enzymes. Sorry, everything is a protein. Okay, what are the exceptions then? Okay, what are the exception? The exception is a ribozyme. Okay, ribozyme is not a protein. Ribozyme and ribonuclease P and peptidyl transferase are three non-protein enzymes. So these are not made up of the proteins, but these are made up of the RNA molecule or with the RNA molecules with combination with the some other molecules, okay, like phosphates or with the some other kind of the substances. Okay, so remember, most of the enzymes are protein. Exceptions are ribozyme, ribonuclease P, and peptidyl transferase. So ribozyme, we will learn about ribosome in detail in the 12th class, okay, especially in the molecular chapter. Here we only come to know that, okay, ribozyme is there. This also act like an enzyme, but these are not belongs to the proteins, okay. So these are the basic things. So we will move on to the next one. Why it will be helpful? Why this enzymes will be helpful? Enzyme will help in the transfer of the chemical reaction. Okay, so is it create any chemical reaction? No. Enzymes don't have the capacity to create a chemical reaction. Okay, I will simply state one idea. Okay, so think that starch is sorry, a glucose is there. Okay, and the enzyme can't convert glucose into something like cyanide, okay, if it is not happening in nature. Okay, it can't convert like this. You think in another reaction, glucose is converting into CO2 plus H2O, but this process is happening in slow. Okay, it's a slow reaction. Then this enzyme and come here, which can speed up the reaction. That means what? The enzymes never create a reaction or never do a reaction or never um, uh, create or sustain a reaction, but it can speed up the reaction. Okay, that means what? These are only helpers. This can't take any decision. Okay, these are enzymes are not kings. These are only helpers. King is running its state, right? King is running his state. But what these ministers and everyone do, this minister and everyone help the kings to run his state, right? Minister will give the idea, minister will help in other things, or he will makes the works to do in a proper time, exactly like that. The reactions are like kings, they will be going on, okay, but enzymes are like ministers, so they will help the chemical reaction go in a much speeder way, much high speed way. Okay, so what we can see here is the glycolysis process. From what you normally get the energy. Okay, step is simple. The glucose should convert into a pyruvate, okay, or pyruvic acid. Okay, simple step. Glucose is there and it should convert into pyruvate. But if you leave it without enzymes, it will take several months, okay, several months. But every second we need hundreds of reaction within a single cell have to convert glucose to the pyruvate, because this reaction will give us the energy molecule like ATP and NADH. Okay, there where actually enzymes are coming. Look how many enzymes are here: hexokinase, phosphoglucose isomerase, phosphofructokinase, aldolase, tryptose phosphate isomerase, glyceraldehyde phosphate dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, pyruvate kinase. Okay, so many enzymes are involved, and they convert complete this reaction, which may take the thousands of hours into a few seconds. Okay, so for this we also have another one example. The reaction, the best example we can take for this one is the highest speeded one reaction. Okay, so that one is the carboxylase anhydrogenase, right? What happens in a regular, one second, yeah, right. Yes, exactly. We will see that how the enzyme nomenclature is there. We will see that in the next one. Okay, in the nomenclature of the enzymes. Okay, we will see it in the nomenclature in the enzymes. So we are taking a simple example, something like this one, carbonic anhydrase. Okay, what it will do? The carbonic anhydrase converts CO2 and H2O into the H3CO4. Okay, H3CO, sorry, H2CO3, not H2CO4, H2CO3. That means carbonic acid. Okay, so what happens in nature? For in nature, conversion of this reaction happen in like 200 molecule per hour. Okay, per hour only 200 molecule is happening. Okay, but if we take an example of carbonic anhydrase, carbonic anhydrase. How it will convert then? This will convert 600,000 molecules, 600,000 molecule per second. Just imagine the strength. Regularly 200 molecule per hour is happening, but as we added an enzyme, it increased to 600,000 molecule per second. That means a particular without enzyme, it will take how many time? It will take hundreds of days, but that will be done in seconds, right? That can be done in a few seconds. So that is the ability of our enzymes. Okay, that is the ability of the enzymes. That much like helpful it is, right? So that much quick it is. Okay, so speeder than a light, ah? No, no, like nothing is speeder than the light, you know that, right? So this is the conversion rate, not its speed, right? It's not like, huh? Both of the things are not same. Probably you guys already learned in the first chapter, shall told, right? We can't compare the things, huh? But I understand this, this one, okay? So that much quicker it is, okay? That much quicker it is. So that is how the enzymes are helpful, okay? So probably as you guys already observed, the enzymes name, okay, how the name came from them, or I can simply put it as the nomenclature. I will write it in here only. Okay, how the nomenclature or naming of the enzymes came then. The nomenclature is simple. The nomenclature may be referring to what? It may be referring to the substrate it act. Okay, it may be referring to substrate it act on. Okay, I can take an example of simple here. Okay, fructose 6 phosphate worked on by phosphofructokinase. Okay, from where it got its name, got its name from this substance. Or you can simply say another one like, so which one we will take? Sucrase or maltase. Okay, maltase is an enzyme. Okay, maltase is an enzyme which act on the maltose. Okay, so from the substrate it's getting the name. Okay, sometime it also get the name from the chemical reaction. Okay, from the chemical reaction which is actually doing. Okay, so for that example I can simply take something like dehydrogenase. Okay, dehydrogenase. So you can I understand this, right? Dehydrogenase means what? Or decarboxylase. Decarboxylase means what? It is removing the carboxylase mean what? It will removing the CO2. Remove the CO2. So that is how it got the name. But remember, whenever we are giving the name, the one thing is important, that is the suffix we are adding. Okay, what suffix we are adding? The suffix we normally add is the ASC normally. Okay, ASE. I'm writing in a bold, but you just imagine that ASC. So whatever example you take here, you can take example here only, right? Hexokinase, isomerase, aldolase, and hydrogenase, right? Kinase, mutase, right? Whenever you take, it will end with the suffix ASC. That is very important. Whenever you heard a word with ASC ending, just imagine it is the enzyme. Just imagine it's enzyme. Okay, so yeah, next one will be the enzyme structure. Okay, how the enzyme structure is there? This is simple. Okay, this is very simple. The enzyme, I can write it something like this, but it is not always like this. Okay, the enzyme is made up of the protein, we know that. Okay, but this is not an active form always. Okay, so sometimes what happens, it needs something else to make it a activated form. Okay, so those proteins, without those portion, we call it as the apoenzyme, or in a simple words I can put something like, this is the organic or protein base, protein base of the enzyme, and it should be attached with something. Okay, that something is the cofactor. Okay, cofactor means what? Cofactor means any substances which attachment will leads to the activation of the enzyme. Okay, any substances will attachment or attaching substances, we can say attaching substances which leads to the activation of the enzyme. When you add both apoenzyme and coenzyme, you will get the holoenzyme. Holoenzyme means whole enzyme. That means a complete enzyme, we can say. How we can say? Holoenzyme means a whole enzyme, or you can say a complete enzyme. Okay, remember what is apoenzyme? The apoenzyme means only the protein part of the enzyme, okay, or you can say simple part of the enzyme. And cofactor means what? It's attaching substances, then both of them, and we will form the holoenzymes. Okay, remember, this apoenzyme is inactive not every time. Okay, I will write down that also. Okay, so if apoenzyme is active, apoenzyme is only active, then we call this particular kind of the enzymes as simple enzymes. Okay, simple enzyme. That means it don't need anything else. Only the protein part is enough. Okay, protein part is enough. Example, I can write something like pepsin. Okay, pepsin. But sometimes what happens? It needs certain things, right? Apoenzyme, if it needs a cofactor, okay, then what it will become? Then it will become the holoenzyme. For what? For the activation. Then we call these kind of enzymes as the conjugated enzymes, as we seen in the conjugated lipids, right? Exactly that kind, conjugated enzymes. So if I take there are like several examples are there, we will see the example later on. Okay, so there are like we can take an example of oxidoreductase enzymes or nitrogenous enzymes. Okay, what is cofactor is made up of? We'll see. Okay, what is the cofactor? This cofactor is again divided into three types actually. One as the prosthetic group, another as the coenzyme, and another one as the metallic ions. Okay, we will learn those things. What are those things? How it will be different? Everything in the tomorrow's class. Okay, so I will repeat this one thing again. Enzyme structure in the tomorrow's class. It is also in the tomorrow's class. Just watch it. Okay, if you don't understand, no need to understand. Pepsin is always no, like Sachin, no, like pepsin also may come the, remember, before this apoenzyme is the proenzyme. Okay, proenzyme. This proenzyme may be become within a particular temperature, that means in a 35 degrees Celsius, may become directly become a simple enzyme and form active. Okay, so that's the, I will write down here, it's also proenzyme to this apoenzyme. Okay, it's active, or it need additional one thing apart from the physical factors. Physical and chemical factors means temperature and the pH, right? If it needs some other components, then it will be conjugated enzyme. Okay, so I will repeat this one more time in next class also. Okay, don't write down this, don't write down this glycolysis pathway, it's not important. We will see that in the plant physiology. Okay, so one more things, guys. Tomorrow is the day after tomorrow is the last class. Exactly, last class. Okay, so relating to this, so in that, if you have any doubt, you can ask. Okay, so if you have any doubt regarding the biomolecule chapter, and another one thing, like Friday someone else will take the class. Okay, this diagram conjugated enzyme, no, you can't say based on the reaction. You have to see a particular enzyme. You have to take a hexokinase, and you have to see the structure of the hexokinase, then you can decide it is simple enzyme or conjugated enzymes. Do you have the test? No, you don't have the test this week. Last class video not uploaded. Oh, I will check. Ha, Sachin, what we have to do? Everyone is pushing us. uh, Amit, 1 second. I forgot that. I'm just checking what we said. Yeah. So then, if you have any doubt, YouTube, just you know how to contact me. Okay. Then I will close the YouTube live chat. Bye bye. Okay. We'll meet day after tomorrow.